Here is the topic basics of gene cloning. Defining cloning. Cloning is a loaded term that can be used to mean very different things. Cutting a piece of DNA from one organism and inserting it into a vector where it can be replicated by a host organism, sometimes called subcloning because only part of the organism's DNA is being cloned. Using nuclear DNA from one organism to create a second organism with the same nuclear DNA. Restriction enzymes, also called restriction endonucleases, are proteins that cleave DNA molecules at specific sites, producing discrete fragments of DNA. Restriction enzymes were first isolated by Nathans and Smith in 1970. Why restriction enzymes? Why would bacterial cells contain proteins that cleave DNA at specific sequences? Generally, restriction enzymes are thought to protect bacterial cells from phage, bacterial virus infection. Bacterial cells that contain restriction enzymes can cut up invasive viral DNA without damaging their own DNA. Joining DNA fragments. In 1972, Paul Berg and colleagues made their first artificial recombinant DNA molecule, demonstrated that the DNA of Seaman virus 40 could be linearized by EcoR1, created a circular dimer of Seaman virus DNA by joining two linearized fragments, also inserted pieces of lambda phage DNA in linearized Seaman 40 virus molecule. Isolating genes, Herbert Boyer and Stanley Cohen built on work of Berg, Nathans and Smith to use restriction enzymes to isolate a single gene, placed it onto a plasmid vector. Bacterial cells were then transposed with the recombinant plasmid. The bacteria host cells replicated the plasmid, producing many copies of genes, thus amplifying it. The practical application was the expensive human protein products like insulin, which were used to treat disease, could eventually be produced from recombinant molecules in the laboratory using bacteria or other hosts. Human protein products like insulin could be used in very large quantities from the recombinant molecule. Patients no longer had to use insulin isolated from pigs or cows. Plasmid vectors. Plasmids are circular pieces of DNA found naturally in bacteria. Plasmids can carry antibiotic resistance genes, genes for receptors, toxins, or other proteins. Plasmids replicate separately from the genome of the organism. Plasmids can be engineered to be useful cloning vectors. Plasmid vectors can be designed with a variety of features: antibiotic resistance, calorimetric makers, strong or weak promoters for driving expression of a protein. Now, here is a diagram related to antibiotic resistant makers. Then, this consists of multi-cloning region. Cloning of a piece of DNA is done in this method following this process. Performing the restriction digest. You will need to set up a restriction digest of your plasmid vector and your DNA of interest. Restriction enzymes all have specific conditions under which they work best. Some of the conditions that must be considered when performing restriction digestion are temperature, salt concentration, and the purity of DNA. Purify your DNA fragments. The insert of interest that you want to clone into your plasmid needs to be separated from the other DNA. You can separate your fragment using gel electrophoresis. You can purify the DNA from the gel by cutting the band out of the gel and then using a variety of techniques to separate the DNA from the gel matrix. Ligation. Ligation is the process of joining two pieces of DNA from different sources together to the formation of a covalent bond. DNA ligase is an enzyme used to catalyze these reactions. DNA ligation requires ATP. Transforming bacteria. After you create your new plasmid construct that contains your insert of interest, you will need to insert it onto a bacterial host cell so that it can be replicated. The process of introducing the foreign DNA into the bacterial cell is called transformation. Competent host cells. Not every bacterial cell is able to take up plasmid DNA. Bacterial cells that can take up to DNA from the environment are said to be competent, can treat cells to increase the livelihood that DNA will be taken up. Two methods of transforming are heat shock and electroporation. Selecting for transformants. The transformed bacterial cells are grown on selective media to select for cells that took up plasmid. For blue-white selection to determine if the plasmid contains an insert, the transformants are grown on plates containing X-galactose and IPTG.